Hello, good morning, everyone. Thank you very much for coming today to our session. So I'm really pleased to be with Emily and Anne-Sophie from the Fluidigm team, and with Senan and Miguel, who work at the EPFL. So just before we start, and while we wait for everybody to log in, I have a couple of questions to ask you. So I'm just going to launch those questions. So do you have any experience with mass cytometry or imaging mass cytometry? So yes, Helios, yes, Hyperion, yes, both, or no, neither. So you just need to click the correct answer on your screen. For the people who just logged in, you just need to click the right answer on your screen, please. I'll give you 10 more seconds. Okay, thank you. And the second question is, what range of markers are you interested in looking at? So is it 1 to 10, 11 to 20, 21 to 30, 31 to 40, or 40 plus? I give you a few more seconds. So if you just logged in, please click the answer that applies to you on the screen, please. Okay, I give you a few more seconds. Excellent, thank you very much. So I'm just going to quickly share the results with you all. So we have 17% here who have experience with Helios, and the rest don't have any experience yet. So I hope you find this webinar very interesting. And for the range of markers, it's a mix. Actually, we have some people 1 to 10, 11 to 20, another 29% for 21 to 30, and 14% for 31 to 40 markers, which is great. All right, thank you very much for answering those questions. So just before we actually get started, I just would like to let you know that you can use the question box, which is on the right-hand side of your screen. So if you have any questions during the webinar, you can type them in there. And what's going to happen is we're going to have a short introduction by Senan, as well as, and then followed by the presentation of Emily. And after that, we will come back to have a Q&A session, and I will ask them all your questions so everybody can listen to the answers. So feel free to type your questions here as we go along, and we will pick those up later. And if you have any issues also during the webinar, please type them in there, and I will, try to help you. So yes, I think I will let Anne-Sophie and Miguel do a very short introductions of themselves, and yeah, have a great session, everybody.
Okay, so good morning, everyone. So I'm Anne-Sophie. I'm the sales manager for Fluidigm, and I'm happy to see all of you here today. So as Cecile said, if you have any questions, please don't hesitate to come back to us, and we will be happy to help you. So Miguel, please.
Thank you, Anne-Sophie. Hello, everybody. My name is Miguel. I'm in charge of the Flow core facility here at the EPFL. It's really my pleasure to welcome you to this webinar done in collaboration with Fluidigm. Thank you so much, Fluidigm, to collaborate with us since a few years now. And I leave now Senan to introduce the facility and also the service of mass cytometry here at the EPFL, and after the presentation of Emily.
So, hello everyone. I'm still on the poll. Can you charge the slide, please?
Emily, you need to go to share your screen and put it back to.
Okay. Normally it's automatically. I'm already sharing my screen.
Perfect.
Yes, perfect.
Okay.
So thank you very much for joining us today. I will not hold the microphone for very long. For people that do not know where we are located within the School of Life Science at EPFL, so at the Lake campus. We are a platform that is working with bioengineers, cancer biologists, neuroscientists, and with the Global Health Institute. So at EPFL, we have mostly collaboration with those people that are really pushing the boundaries of science forward. And thank you very much to all of them. But also we have collaborations. With some people at the University Hospital in Chuve, in Lausanne, with the University of Lausanne, with the University Hospital of Geneva, and some outside collaboration, notably in Italy. And with them, we work with human samples. So we have both collections, mice internally and mostly human externally. So if you are wondering how to approach us, it's actually very simple. You just contact us via email at CyTOF@epfl.ch. We do a first meeting where you will present your project. We will do a feasibility check, biosafety approval, and then we are good to go in order to design the panel. We already have a collection of 500 antibodies, both in mice and human. So we have ready-to-use panels, but also we can design tailor-made panels for you. Last but not least, we have a stable collaboration with a Bioinformatics Competence Center at EPFL and UNIL for very complicated bioinformatic analysis. But we have also our own pipeline in order to generate an unbiased high-dimensional analysis. So now I will let the mic to Emily. Emily, thank you very much for sharing some of your knowledge with us.
Okay. So yes, so many thanks to all of you to be there today, and thanks to Sena and Miguel to host them today. So I am Emily. I am field application specialist for Fluidine, and I am involved on all the mass cytometry application and everything. So if you need some helps, you can contact me directly, and we are in contact with the EPFL, also the core facility. So do not hesitate to contact us. So what we will see today, we will have a really quick introduction, because a lot of you are already using the Helios, so I will try to do that really briefly. But for those who are new, we will look at how it works and how to start with the mass cytometry. And after that, we will look at what's new, what are the last innovation we launched this year, and we will see really quickly some publication on mice model, because you are like a mouse facility. So yeah, and I will introduce you really briefly the imaging mass cytometry. So to start, you know that when we work with the immune cells, we have a lot of heterogeneity in these cells, and we have a lot of different cells like DC, B cells, T cells, etc. And for all of these cells, we can visualize some markers. So for example, here, I will just put my laser. Okay, so now you can see it normally. So for the B cells, for example, you can have a lot of markers. For T cells also, you have dedicated markers for all of these different cell types. But if you want to have more markers, for example, functional markers, cytokine markers, phosphor markers, or transcription factors, it's also possible to look at the function of the cells. And this is possible with the flow cytometry to look at the single cell level and to look at the markers expressed by these cells. And we know that there is some limits of the flow cytometry, like the fluorescence spillover with spectral overlap, for example. We have also high variability between the different intensity of the signal. We can have up to 1 log of difference between 2 fluorochromes, for example. And you know that fluorochromes are like really sensible to many parameters, like the temperature or the light, for example. And everything here with the flow cytometry, all the signal detected is light signals, so the autofluorescence of the cell can interfere with the signal of the antibody that we use. So to go over all these limitations, the mass cytometry was developed. And so now with the Helios, the last version of the instrument, and the Helios is the one you have in the EPFL, is the CyTOF 2 upgraded in Helios, so it has all the characteristic of the Helios. You can now analyze more than 50 parameters in the same time on just one tube. So this is possible by using antibodies labeled with metal tags, so isotopes, pure isotopes. And as you can see here, we can have this separate and distinct signal with no overlap of the spectrum. So we have really clear signal, and we have less variability between the intensity of our signal. We never have 1 log of difference between 2 isotopes. And I would say, last but not least, we don't take into account the autofluorescence of the cells, because here the signal integrated is a mass of the isotope and not the light of the fluorochromes. So we don't care if our cells are autofluorescent naturally, or if it's an induced autofluorescence, for example, when we look at into intracellular markers. So you can see here that there is a lot of free space available, so we hope to have more metals in the future. And just for the new people that doesn't know, the metals that we are using are non-rare, so they are present in the Earth crust, so it's easier to extract them and use them, and they are non-biological, so thanks to that, we will not have interference between the isotope used for the staining and isotopes naturally into the cells, and all the metals that we are using are non-radioactive. So you can use them at the bench without any specific dispositive or something like this. And now we will see how it works. So as I said before, mass cytometry is cytometry first of all. So you start with your sample, and of course your sample is stained with metal tag antibodies, metal labeled antibodies, and not fluorochromes labeled antibodies, and then you introduce your sample into the instrument. So here you have the nebulizer that will create a single cell stream, and you will have a creation of a lot of droplets, and each of these droplets will contain 1 cell. And each of these cells will go through the argon plasma. And the argon plasma is like a fireball, and all the plasma will be automatically ionized. So each cell will become an ion cloud. So you have here a cells, and after that you have an ion cloud. Something that you have to know is the temperature of the plasma is around 5,500 degree Kelvin, and it's the temperature of the sun surface. And it's why we call this instrument the Helios. So when we have this ion cloud, we have to clean the ion cloud, and it goes through this quadrupole, and we remove all the isotopes with a mass less than 75 Dalton. So we will remove all the ion that constitute the cells, like carbon, oxygen, nitrogen, and things like this. I will just wait 2 seconds. Okay. And when we have this clean ion cloud, we send all this clean ion cloud into the TOF chamber. So this is the detection part of the instrument, and we will just send all the ions into a time of flight chamber to separate the ion of the cloud, and all the small ion will arrive first, and the big one will arrive after that. And all the signal that hit the detector here will be integrated in the files, and it's a FCS file, a classic file that you can use with analysis software. So how can we start with mass cytometry? So the first thing to do is design the panel. So as Senan said, they can help you to design the panel because it's an extremely important step. And for that, we have some metal conjugated antibodies in our catalog, so more than 750 antibodies. But if you want an antibody that we don't have into our catalogs, it's also possible to label your own antibody. So we are offering some labeling kits, so it's also possible. You can see with Senna and Miguel if you want to develop these kind of markers, and they can help you to do that. And another thing that you can do is using our panel kit. So our panel kit is our kits developed to facilitate your project. So we already designed the panel for you. We include all the buffers and all the protocols into these kits, and you can customize this panel. So I will show you a brief example of our MaxPar mouse spleen and lymph node phenotyping panel kit. So you can see here we have a kit with 17 markers, and these 17 markers allow you to study the main population of the immune cells into mouse spleen and mouse lymph node, but whatever you want. And this panel can be a backbone for your future panel if you want. So you can use other panel kits to mix and match. For example, this one for the phospho panel kit, or this one dedicated on the cell cycle study. And whatever the one you choose, you can see that on the wheel there are a lot of remaining free channels, so you can add any target you want to this backbone panel. So if you are interested in a specific type of cells, you can just add more markers. So if you are interested in NK cells, for example, you can add a lot of NK markers in these free channels. So now we will see what's new with the mass cytometry and what are the new regions. So the first recent innovation that I want to present you today is our MaxPar Direct Immune Profiling assay, and it's an evolution of our panel kit. The panel kit I showed you before. So this kit is dedicated to human samples and is composed by one tube containing 30 markers, but 30 dried antibodies. So all the antibodies are already dried into the tubes. So with these 30 markers, you can analyze 37 population, and this is possible with a software that we propose with this kit. And thanks to that, all the process is standardized from the staining of your samples. So you just have to put your samples in the tube. You can use PBMCs or whole blood. It's validated for human samples only for now. And then you can acquire the samples and generate a report. You can generate this report automatically, and it takes only 5 minutes to do that. And at the end, you will have all your statistics in a PDF report, and you can also extract them in an Excel sheet. So all this process is standardized because you limit the impact of the users since the staining, so you don't have to prepare the cocktail to the analysis because you don't have any bias due to the manual gating. So this kit is composed on 13 markers, as I said before. You have the list here, and you can see that there is some remaining channels. So there are 4 available channels in which you can put additional markers. So you can choose these markers in our catalog. You can do a custom antibody if you don't find your markers in our catalog, or you can use our expansion panel. So here we have developed 6 expansion panel that you can mix and match with the MaxPar Direct Immune Profiling assay, and these 6 expansion panel are dedicating to some population like T cells, NK cells, or myeloid and B cells. So to assess the robustness of our kit, we ran a study across different sites. And in this study, we acquired several samples, so whole blood and PBMCs that were stained with our MaxPar Direct Immune Profiling assay. So we acquire these samples across 6 and 7 sites in North America. And as you can see in this graph, we see that there is a high reproducibility, a high intersite and high intrasite reproducibility. And to go further, we thought that freezing stained samples should be an interesting possibility to allow multi-site projects, for example, or long-term projects. And in this context, we developed an easy protocol to fix and freeze stained samples. So here you can see our app notes. So if you want to have more information, this note is available on our website, and it includes all the tests we run to assess the stability of the cryopreservation. So as you can see here, we have stable results for 4 months. And all these study and these kits allow to develop new working methods like this one. So in this workflow, you can see that we have steps here that is a freezing steps, stain sample freezing steps. And thanks to that, you can send your samples to a core facility that have the instrument. So it's don't need to have an instrument on site. You can collect your samples, you can stain them, you can fix and freeze them, and then you can ship your samples to a core facility that have the instrument, and it makes all the process easier. So this was all the information I wanted to show you about this kit, this new kit that is the MaxPar Direct Immune Profiling Assay. And now I want to talk to you about another range of projects, and these projects are the barcoding. So for those that don't know what is the barcoding is a process to stain specifically your samples, different samples, with a dedicated tag. And after that, you can pull all of them all together into a single tube. So then all the process you will apply to the tube will be the same for all the samples. And we are currently proposing two options for barcoding. The ancestral one, I would say, is the palladium barcoding. It's not novelty, it's something used and a lot with users of mass cytometry. So the palladium barcoding use palladium loaded polymers that stain all the proteins, and to have a suitable staining, you need to fix cells prior barcoding. And this allows the polymer to goes into the cells and stain all the protein into the cells. So these kits have strong advantage because it can stain all the cells, whatever they type. So you can stain immune cells, tumor cells, whatever, and you can pull up to 20 samples in the same time. The second option is the new one, and this is the live cell barcoding. So here the principle is the same. So you encode your sample with a unique barcode, and after that you can pull them into one tube. So the difference here is that we propose to use CD45 conjugated to cadmiums, and with this type of barcoding you can encode up to 35 samples, 35 different samples of immune cells, and you don't need to fix your cells prior to the barcoding. So if you want to have more information, we presented a poster at CyTOF 2020, and you can find it on our website. And the last new project we launched this year is our new panel designer. So this tool is a software to help you to design your panel. So normally you will work with Senna maybe to design your panel, but you have to know that we have this tool that help to design panels. So for those that already using our technology, you may use this software, and just be aware that we have a new version of it now, and this new version is more complete. So now we can have access, you can have access to all of our metals for the custom antibodies, including the cadmiums. And it's also possible to import personal catalogs and to add new metals thanks to the Manage Metals section. So as you can see, Fluidigm invests a lot in research and development to bring new project, new process, and innovation. And all these works allow to democratize the technology, and we have more and more publication now. So as you can see here, there is a constant increase of the publication each year since the beginning in 2014. And we have now more than 1,000 of publication mentioning the mass cytometry. And it is not only publication that are increasing, but also clinical studies. So we have now more than 100 of clinical studies running, and this number increased even faster with the launch of our MaxPar Direct Immune Profiling Assay, because now it's super easy to use mass cytometry, and it's super robust to do mass cytometry. So all these publications cover a wide range of applications. So for example, oncology, immunology, allergy, infectious disease, and so on. And it's important to note that majority of the publications are about big panels. So with mass cytometry, we use mass cytometry for big panels. So more than the half of the publication in 2019 used more than 30 markers, and it's even more now. So you can easily find all the publication mentioning the mass cytometry on our dedicated webpage. So now I will present you 3 papers on my studies using the mass cytometry. So the first one was published in 2018 from a collaboration between teams in Singapore and Melbourne. And they studied here the maturation stages of neutrophils from mice bone marrow, and they redefined neutrophils precursor identity. So they do that by analyzing proliferating neutrophils lineage. And as I said before, mass cytometry is a strong tool to analyze small samples. In fact, here you can put a lot of markers into just one tube. You don't need to do several panels, and it's super important for precious samples like bone marrow samples, for example. The second paper I want to show you is more neurology-oriented paper, and this team here developed a 43 markers panel to study the immune cells involved in the central nervous system of mice. And thanks to this panel, they help to deeper characterize the central nervous system associated disease, dendritic cells. So as you may know, microglia is highly autofluorescent, and here the mass cytometry allowed to analyze all the cells involved in the central nervous system without any interference of this autofluorescence. So this is possible because we are looking at the mass of the isotope and not the fluorescence of fluorochromes. And last but not least, this paper is a paper from the Allison lab. So as you may know, James Allison won the Nobel Prize in 2018, I think, and he won the Nobel Prize for his work on immunotherapy. And here in this paper, he assessed the efficacy of mono and dual immunotherapy. So they do that by studying the TILs, the leukocyte infiltrating the tumor in the mice model. And it showed that dual immunotherapy using both anti-PD-1 and anti-CTLA-4 immunotherapy and the mono therapy using anti-CTLA-4 leads to an expansion of activated effector CD8 T cells. And this is not the case for the mono anti-PD-1 therapy. So this is really interesting to go further with immunotherapy and to apply that to maybe human therapy. But we have to be careful because sometimes the composition of the microenvironment is not sufficient. Sometimes we need to have more information and more information about the location of the cells. So this is what is showed in this picture. So it's important to have the information that we have immune cells close to the tumor, but we have to know where these cells are located. So in this case, we can have different type of tumor, hot inflamed tumor with a lot of cells into the tumor, cold excluded tumor with cells, immune cells in the periphery or really close to the tumor, and cold ignored tumor with no immune cells. And this is why we developed the imaging mass cytometry. So I will just show you really quickly the imaging mass cytometry, and will explain you what is it. So here you can see the Helios in the background, and you can see this module, and all of these compose the Hyperion. So this module is an imaging module that you can connect to the Helios, and this allows you to do imaging studies. So you can now study more than 30 markers in just one slide, and it's super easy because the main protocols are classic protocols that you can have with IF histology, for example, or things like this. And it's super important because you can have all this information of the location of your cells, not only the composition of your tissue, but also the location of the cells into your tissue. So it's really powerful because you can put all these 30 markers in one slide, and you don't have to do a lot of slides to have all this information. And it's brand new things because with classical histology procedure, you cannot have this amount of information. And to do that, we are still using antibody tagged with some isotopes. So it's exactly the same process than before. And the main advantage is the fact that we don't have any autofluorescence. So here you can see results from IF staining, and here IMC staining, imaging mass cytometry staining, and you can see that we don't have any autofluorescence signal when we are doing imaging mass cytometry. So the workflow is really easy. You just have to design your panel. It's the same thing for the mass cytometry. It's the first thing to do. Choose all the markers you want to use, and then stain your tissue. So here the protocol are really close to IF immunofluorescence protocol. So you just have to stain with your antibody labeled with your isotope. So it's a primary staining, a classical one, and then you acquire the slide with the Hyperion, and then you can analyze your results. So we have more and more publication with this system, and I will just show you briefly one result, one paper, so published in 2020 this year, and he showed that by using the imaging mass cytometry, they can find new groups to stratify patients with breast cancer, for example. So we can go further because with the actual technology, sometimes we can have some limitation. And here they studied more than 300 patients, and they revealed 18 new groups to stratify the patient suffering from breast cancers. And yes, another paper just to show you that it's possible to go over the 30 markers. So this paper used a 14 markers panel for IMC, imaging mass cytometry, and they explain how they did that in this paper. So many thanks for your attention, and please be free to ask all the questions you have in the question box, and Sena and I will answer them. Many thanks.
Thank you very much, Emily. That was a great presentation. So very luckily, we got some good questions from the audience. So I saw the new Max Direct kit only works on Helios. Is it true?
Yeah, Yes, it's dedicated to analyze with the Helios, and it's also only for human samples. But yes, you can use it with the Helios only.
Excellent. So is the panel designer working for Hyperion panels?
No, it's quite different with the Hyperion, because the panel design of Hyperion panel is even simple. So you don't have any oxidation, so you have less protons with Hyperion than with Helios. So you will not have to use the panel designer to do that. Just contact us and we can design the panel with you. But for now, the panel designer is not done to do panel design for Hyperion.
Thank you. And I've got another question from Jessica. So do some antibodies fail to stain after coupling with metals?
Normally, no. So you have to strictly follow the protocols. So the protocols, you have to add the polymer. So how it works? So you have a metal, you have a polymer, and you have your antibody. You have to put your metal into your polymer and then link your polymer to your antibody. And this is done into the SH remaining part of the antibody. So you do a little reduction and you just link the polymer to this SH group. So if you strictly follow the protocol, it should be perfectly okay. You just have to be careful to not reduce too much your antibody. But if you follow the protocol, it's okay. You will not have any problems. So if your clone is working in flow cytometry, it should work in mass cytometry. After that, it's depending on the clone of the antibody, but maybe Zena can say something about that. But normally we have a good proportion of antibody working and really few are not working. You cannot, for example, label IgM because of the structure of the antibody, but for all of the others, it's okay.
Excellent. Thank you very much. Another question, can I add intracellular markers to this kit?
To the MaxPar kit, I guess. So yes, yeah. It's possible to, yes, it's possible to add intracellular markers. You can put what you want. So intracellular markers is not an issue with mass cytometry because you don't have any problem with the stability of the isotope. So you can use the regions you want to permeabilize, and yeah, you don't have any autofluorescence issue. So yes, you can do that.
Okay, great. And final question. So if you have any questions, please, that's the moment. Can I still use barcoding?
With the MaxPar directive, so we will have to discuss that. So if you use the palladium barcoding, the palladium barcoding needs a fixation, so it's not recommended to do a barcoding prior the MaxPar direct immune profiling assay, but you can do it at the end just to standardize the acquisition. And you can use the live cell barcoding if you want, but be careful because you cannot stain more than 3 million of cells with the MaxPar direct immune profiling assay. So it's, yeah, you can maybe barcode 3 samples, but not more because you need to have enough cells of each of the samples.
Great. Thank you very much, Emily. So I think that's all the questions for today. I just would like to let you know that there are still some, spaces for you to book yourself in with Emily if you would like to discuss your project, on a one-to-one basis, or you can just email her. So you can see emily.gregory@fluidigm.com if you have any questions. And what's going to happen now is I'm going to send you all the recording of this session. So bless you. Feel free to share it with anybody that might be interested in the subject, or rewatch it if you really loved it. Yeah, with that, I just would like to thank everybody for coming along, and thank you so much, Emily, for this great presentation, and for Miguel, and a big thank you to Miguel and Senan, who really helped, with the preparation of this webinar today. So thank you so much, and have a great day, everybody.
Many thanks. Bye.
Thank you. Bye-bye.
Thank you.